All right, so we are on to the next structure function analysis, and we're going to be looking at hemoglobin. Okay, the function of hemoglobin in our bodies is to transport oxygen from our lungs that we breathe in to our fingertips, right, to all the muscles in our tissue. Okay, recall that oxygen is necessary for cellular respiration to occur. So in order for our cells to generate energy, they need oxygen to do that. And if you think back hard enough to biology, you should have learned that oxygen is the terminal electron acceptor of the electron transport chain. And that ultimately drives the formation of ATP within our cells. So that's why we need oxygen. Okay. All right, so let's look at the structure of hemoglobin. Here's another one of those images from David Goodsell, and this just gives you an idea of kind of what a red blood cell really looks like. So here, this kind of purple is showing the membrane. So we've got that lipid membrane here. Here's the outside of the cell. So this is the stuff that's in our blood. Our blood has antibodies in it. It's got some macrophages in it. It's got nutrients pumping in it, glucose molecules, other small proteins, hormones, all kinds of stuff is found in our blood, right? But so inside the cell here, you can see all of these red kind of four-sided clover-looking things are hemoglobin molecules. Okay, so those are all the proteins. So one red blood cell is packed full of hemoglobin molecules. So let's give you some statistics here. 300 million molecules of hemoglobin are found in each red blood cell. 300 million. That's 3E8. If that's hard to remember, that's also the speed of light. But so 300 million hemoglobin in one red blood cell. Okay, each hemoglobin, we'll learn, can carry 4 molecules of oxygen at any given time, up to 4. Okay. 2.5 liters is the total volume of red blood cells in the average 150 to 180 pound human being. The total amount of liquid blood there is about 5 liters. So half of your blood is made up of red blood cells, which each one red blood cell is carrying, 3E8 molecules of hemoglobin. And each one hemoglobin can carry 4 oxygen molecules. So you can see how immediately we're starting to talk about a lot of molecules of oxygen pumping around in your blood. 90 femtoliters. Remember, femto is e to the negative 15. 90 femtoliters is the volume of one red blood cell. So you could, from those 3 numbers alone, calculate how many total oxygen molecules all of the hemoglobin in the average adult could carry, right? So you know the total volume of red blood cells, 2.5 liters. You'd figure out how many total red blood cells there are by dividing out 90 femtoliters into that, right? One red blood cell is 90 femtoliters. So if you take those 2 numbers and work your dimensional analysis, you should be able to figure out how many total red blood cells there are in the average adult. Then you would take that number and multiply it by how many molecules of hemoglobin there are in one red blood cell. And that'll give you the total number of molecules of the hemoglobin protein. Then you could multiply that number by 4 because each one hemoglobin can carry up to 4 molecules of oxygen. So, I might make that a test question and maybe ask how many grams of hemoglobin what's the maximum amount of mass the average human can carry in oxygen just solely due to hemoglobin. So those numbers would be important for you. Okay, some more facts about red blood cells. 2.4 million red blood cells per second. So your body is making 2.4 million red blood cells every second. So some red blood cells are being taken off the market and other ones are being pumped in, right? So that's about how long it takes, or how many red blood cells your body's making. Remember, that's coming from your bone marrow. Takes about 60 seconds for one red blood cell to travel and circulate through your entire body from heart to heart, or lungs to lungs, if you want to, use that as your starting and stopping point. And each one red blood cell lasts for about 3 months before it has kind of worn down enough and gets absorbed and eaten by macrophages. But remember, like I said, you've got constantly an influx in and an influx out. Some are being taken off the market, and other new red blood cells are being generated by your bone marrow and pumping out into your blood. The human body is fascinating, and biochemistry is really fun because you really get to understand the chemistry of everything and how it works. I digress. Okay, back to hemoglobin. So, previous structure function analysis of myoglobin, we can overlay or superimpose that structure, which is green here, with the 2 different subunits that are found in hemoglobin. So we've got alpha and beta hemoglobin. And what you notice is that when you superimpose all 3 of those different subunits, they are incredibly structurally similar. Likely they have evolved. The hemoglobin has evolved from myoglobin. I mean, if you want to think about it, probably myoglobin was the first protein, oxygen protein to kind of hit the scene because it was storing oxygen. You put some mutations on the surface of that myoglobin and it keeps mutating and evolving over time, and you get a brand new protein, which is hemoglobin, and it has a brand new function. Now, instead of storing oxygen, it can transport oxygen. Okay, and we're going to definitely learn more about how it does that. So let's look at the structure of this. So like I said, let's kind of do a quick, can't really do primary analysis, but secondary, tertiary, quaternary analysis of hemoglobin. So as you can see, it's a quaternary structure. Hemoglobin is made up of 4 different subunits. I've got 2 of them are colored kind of a light gray, 2 are colored a dark gray, and I've got those heme porphyrin rings highlighted in blue, and this hemoglobin has those 4 oxygen molecules, which are in red spheres, right? So this protein is made up of 2 alpha subunits, 2 beta subunits, and so that would make the quaternary structure of hemoglobin a dimer of dimers, okay? Like I said, it has 4 bound heme groups, so each one subunit still has that hydrophobic cavity where that heme can bind. It's got that iron center, so we can still have the oxygen coordinate with that distal histidine to the iron center, okay? It has a central binding cavity. Okay, it's hard to see now, but when we rotate out, there's going to be this area in the center. Let's see if we can find it. We're kind of in the center of all the 4 subunits, and there's a really good picture coming up. Just give it a second. Right, it's going to be like right here. And there it is. See straight through. Yes, so that central binding cavity is how we're going to modulate the function of hemoglobin through other interactions with other compounds that your body will make. Okay, so it's a big central binding cavity. Okay, and the amino acids that point into that binding cavity, we're going to also look at in a little more depth. Okay. Hemoglobin, given that it has 4 subunits instead of 1 like its monomeric cousin myoglobin, shows cooperative binding. So remember when we saw the shape of the curve? So myoglobin binding, oxygen binding curve has a hyperbolic shape, and hemoglobin will have a sigmoidal shape. So hemoglobin shows cooperative binding. And what cooperative binding means, the definition you should write down and stick up here in memory. Cooperative binding means when one molecule of oxygen binds, that makes it easier for the other molecules of oxygen to bind. And it's cooperating with each other. So if it's fully deoxygenated and you can stick 1 molecule of oxygen on, it might be hard to get that one in, but once you put 1 molecule of oxygen into this bigger structure that has 4 kind of connecting subunits, it makes it easier for the other 4 to bind because it creates little tiny conformational shifts that make that hydrophobic pocket where the heme is a little bit bigger so an oxygen molecule can slip in. Okay, so that's cooperative binding. Okay, here's kind of an picture of what cooperative binding looks like. So really, another way you can think of cooperative binding is that changes in the tertiary structure change the quaternary structure. So changes in tertiary changed quaternary structure. So here I've got in red, so this is the alpha helix that has the proximal histidine. This is the proximal histidine. So remember, the proximal histidine is the one that's in very close proximity to that iron center of the heme group. So this says the porphyrin plane, the really flat, hydrophobic heme plane that's sticking inside that hydrophobic cavity of the different subunits. Okay, so the red is showing oxygenated, there's our oxygen molecule, and the blue is showing the deoxygenated hemoglobin subunit, so no, without oxygen. And what you can see, and you can even see this in the crystal structures, is that without an oxygen bound, this helix that has the proximal histidine on it is in a more kind of relaxed state. So it's shifted just a little bit, and it pulls that iron just out of plane of this of the porphyrin plane of the heme group. So see, the iron is not directly in the center coordinating with those nitrogens. So again, so the tug from the without oxygen being there, so that little tug from the alpha helix kind of pulls the iron out of pocket. Okay, that might be good, because if you remove, like, iron has 26 electron, well, iron 2 doesn't have 26, normal iron has 26. Iron 2 plus is going to have 24 electrons. So there's this electron cloud, you got to remember, that's 24 electrons, that's kind of big around the iron center. So if you get that out of the way, that might make it easier for that oxygen to slide on in there. And once the oxygen does slide on in there, remember, it's going to interact with the iron's going to donate an electron to it. We make a superoxide, an O2 minus, and then that can coordinate with the distal histidine, and that also tugs this alpha helix up a little bit. So notice that this whole helix shifts. I forget what the actual degree is. Maybe it's something like 12 degrees, shifts upwards and pulls that iron back in plane. So when oxygen's bound, the iron is now back in the plane of the porphyrin ring system. So this change in this alpha helix, though, that shift creates a structural change to the other subunits that make it easier for oxygen to bind. Okay. Again, changes in the tertiary. So the tertiary, we're kind of looking at the changes of the tertiary. So we're talking about this histidine and the interaction it has with that iron center. Okay, so we change the tertiary structure, which is that alpha helix, and that's going to change the quaternary structure. If you don't believe me, here we go. Look at this. Here is our 4 subunits of hemoglobin. You can see there, right here. So this red is that heme porphyrin ring. We've got down here our proximal histidine, right? In the center of this, we should have that iron molecule, and here's our oxygen, okay? So this image was created from the crystal structure data we have from the oxy and the deoxy hemoglobin structures. So look at the shift that happens and occurs when this oxygen binds, okay? This is the cooperative binding. This is changes in tertiary. Notice this is the helix that proximal histidine's on, that it shifts. And as it shifts, it changes its interaction with these 2 subunits here, and that creates kind of a big chain effect. So everything shifts a little bit. Okay. So when you talk about cooperative binding, that it means that there's usually 2 different states. So the 2 states for hemoglobin are called the T state and the R state. Okay. The T state, T stands for tense. It's the deoxy state, so without oxygen. And that's when the iron is the iron 2 plus center. And it's also when the iron is slightly out of plane with that porphyrin ring. Okay. Tense, this is how I remember it. The T state is tense. If you're like, oh, really tense, this hemoglobin's really tense and it's saying, where's the oxygen? So it doesn't have any oxygen bound. That's the T state. Like, I don't know where my oxygen is. Where is it? I'm really tense. Conversely, when oxygen is bound, that's called the R state, and the R state's the relaxed state. So maybe you're relaxed because you're like, oh gosh, all right, I can do my job. I've got the oxygen. That's how I remember it. So R state means relaxed. It's also the oxy state, And then that's when the iron center is pulled in plane with a porphyrin ring. And the iron had donated that electron to the oxygen molecule to make that superoxide. And you have an iron 3+ center there. Okay. But so this, again, this GIF or GIF is showing how changes in tertiary structure can change quaternary structure. Okay. Hence cooperative binding. And again, we see it in the curve. So to recap, this is our hemoglobin curve. Our y-axis is fraction of O2 bound. 1 is 100%, 0.5 is 50%. It's fraction. So fraction of O2 bound. Our red line here was the myoglobin. It's hyperbolic. But look, the hemoglobin one is not hyperbolic. It's sigmoidal. And anytime you're looking at binding curves and you see a sigmoidal shape, that indicates that there is cooperativity or cooperative bindings going on, meaning it's going to have multiple subunits, right? Monomer, myoglobin is just a monomer. You see this hyperbolic. It's either kind of almost in an on or off state. But hemoglobin, you have 4 different pockets that you could mess around with. So it's definitely, those 4 subunits can interact with one another slightly differently. And you just get this cooperative sigmoidal shape to the curve. Okay, so remember, I told you about that open big binding cavity. Remember that pi-mol structure was flipping around. I said, there it is. You could like practically see straight through the center where those 4 subunits, well, the 4 subunits kind of come together, but they leave this big hole right there in the gaping center. Okay, so hemoglobin is what we'll call an allosteric protein. This is a new word I want to introduce to you is allosteric. So it comes from, it's Latin or Greek, I don't remember, but the word is allos, meaning other. Okay. Allosteric proteins mean that molecules can bind and kind of hold the protein in one of the 2 states. It can hold it in the other state. That's how the allosteric gets its way into the vocabulary here. Okay, so hemoglobin is an allosteric protein. That means molecules can bind and regulate what state it's going to stick in of the 2 or multiple states. Okay. All right, so the biggest player of that is 2,3-BPG. It's also sometimes said 2,3-DPG. This 2,3 is B for bis, P for phospho, G for glycerate. Okay, glycerate is this backbone carbon, 3 carbons with the carboxylic acid here. 2 and 3 bis phospho. Bis means 2 phosphate. So I've got 1 phosphate on the number 2 carbon, the other phosphate on the number 3 carbon. So 2,3-BPG or 2,3-DPG, 2,3-diphosphoglycerate is the D. This is the molecule that sticks in that center cavity. It can interact and get into that center cavity and stay there. Okay. How does it stay there? What do you see about this molecule particularly? You should notice that it has a lot of negative charges on it. We've got 1 here from this carboxylic acid moiety. We've got 2 minuses from this phosphate and 2 minuses from this phosphate here. So kind of overall, that's a net negative 5 charges. So we just got this really negative small molecule. Okay. If we look at the central cavity, and we're going to definitely do this in pi-mol, and we're going to recreate this picture, but in pi-mol, and you're going to have to label it and show me these interactions, but I digress. Okay, so we've got our 2,3-BPG bound right here. So what side chains is it interacting with? We got to think, this molecule is really negative, so it's probably going to be interacting with really positively charged molecules. So we should see maybe some arginines, some lysines, and some histidines. So what do we see? We see that we've got our, we've got 2 histidines here. So these are histidines. We've got a lysine here. Oh, I think this is the N-terminus. Because look, here's a lysine, so it's got more squiggly. So there's the lysine. We've got an N-terminus of one of these subunits. So the N-terminus also has an amine group, so it's positively charged. Likely there should be symmetry here. So we should see if there's an N-terminus here, there should be an N-terminus over here. We've got 2 histidines here, so we should see 2 histidines down here. We've got a lysine here, which means we should have a lysine on the other side as well. So that cavity, when it is big, opened, because it, as we see in this previous slide, based on if you're in the T or R state, kind of remember that shift can make that cavity bigger or smaller. So when it's in the T state, remember T is tense, T is where's the oxygen? So no oxygen bound. That cavity is kind of big, and this 2,3-BPG fits right in there. You could also say that 2,3-BPG stabilizes the T state because it holds it, right? This cavity can't get smaller because you've got a molecule now stuck there in the center. So you can't switch into this R state because you've got something that's not allowing this to get small again. So you can say that 2,3-BPG, which is an allosteric inhibitor, stabilizes the T state because it sits in that central cavity. Okay. And since the T state is the deoxyformer without oxygen, you would say it's you could call it an inhibitor. Okay. The opposite of an inhibitor would be an activator. It would activate it. So likely if you had a molecule that would stabilize the R state with the oxygen bound, you could call that an allosteric activator. Okay, so 2,3-BPG plays a vital role in releasing oxygen off the hemoglobin. Okay, 2,3-BPG is found in your red blood cells. It's part of it. It's leveled. It's a molecule that's created as a little side reaction from the glycolysis pathway. So, I mean, everything in your body is chemically related. I got this big picture back here. Those are all the metabolic pathways. And right there, that red line, that's glycolysis. And so I probably could go over there and point where 2,3-BPG flies off of that glycolysis breakdown, but that would be more than we need to see. All right, so those are some of this is like a major contributor of the game. This plays a role in helping release the oxygen once it reaches your tissues. And also it plays a role in high altitude training. If you're an athlete and you've ever heard of people training at high altitudes, it's because your body's going to create more 2,3-BPG. And I got a couple slides coming up on that, but that's a little teaser. Okay, so if we look back at the sigmoidal binding curve, so again, remember, this one is just percent saturation. So instead of fraction bound, it's just how saturated the hemoglobins are, 100%, 50%, 10%. And then on the x-axis down here, I've got the pressure of oxygen in millimeters of mercury, which is the same thing as a torr. Okay, so we've got our black line is the normal sigmoidal binding curve, but you can shift this curve. This curve can shift to the left or to the right. And if you recall, when we talked about myoglobin, we said that the partial pressure of oxygen in the lungs is about 100, and the partial pressure in the tissues is about 40. So notice if we were to draw 2 vertical lines at 40 and 100, a shift to the left or a shift to the right can change the percent of oxygen that it gets offloaded into the tissue, right? Because in the lungs, you're pretty much almost 100% saturated. So all 4 subunits have an oxygen bound. And when you get down to the tissue, about 70% of the hemoglobin molecules are saturated. That equates to roughly like 1 oxygen molecule being released while 3 are still kind of stuck on the 4 subunits, roughly. Okay. But you can, so I don't know. So if we've got go from about 100 to about 70, we're offloading roughly 30% of the bound oxygens when the blood travels from the lungs to the tissues. Okay. But you can shift that curve, and that curve shifts for different reasons. And here are some of the reasons. So you can have a left shift. So that's going to make the curve go to the left. And left shifts indicate that the R state, or the oxygenated state, is stabilized. Okay. So what would cause a stabilization of the R state? A decrease in temperature can cause that. A decrease in the amount of 2,3-DPG or BPG. Remember, that's the allosteric inhibitor. Well, yeah, that makes sense because the allosteric inhibitor stabilizes the T state. So if you decrease the concentration of it, you'd be able to get more of your hemoglobin into the R state. a decrease in the concentration of H plus. We'll talk about that more specifically. And another thing that is kind of the left shift, which is happening in me right now, is the fetal hemoglobin. So at one point you were a fetus too, and your hemoglobin binding curve is a left shift. And we'll talk about that. So let's kind of break some of these down. Okay. Oh, the last one is carbon monoxide poisoning. I accidentally overlaid fetal hemoglobin on top of carbon monoxide. So you sometimes hear about people dying from carbon monoxide poisoning. And so why is that? So technically, well, it causes a left shift because it stabilizes the R state, but instead of oxygen molecules being bound, carbon monoxide is bound. And the problem is carbon monoxide binds tighter than the oxygen molecule. So it's less likely to leave the hemoglobin active site area and allow an oxygen to get in there. So you die from carbon monoxide poisoning because you no longer can, these no longer deliver oxygen molecules. But remember, the R state means it's in the oxygenated state. I just want to reiterate that it just appears as though the hemoglobins are in the oxygenated state, but instead of oxygen, they're all carrying carbon monoxide. Okay, remember, carbon monoxide is binding tighter. It's not really going to easily get out of that pocket. So you essentially kind of kill the function of the hemoglobin with the carbon monoxide. So it gives it the look that it's in the R state. Okay, what about the decrease in the H plus concentration? So remember, so if we think back to a decrease in the pH, we got to start thinking, what does that mean? If you decrease the H plus, that means you're getting rid of the H plus, right? So you're increasing the pH, you're becoming more basic. So what happens to some of those? There's only like one residue I'm thinking of particularly that has a pKa around physiological pH, which is histidine, right? pKa of 6. So if you start making the solution more basic by decreasing the amount of H plus, you're going to start deprotonating these histidine residues. And there's 4 of them that are right there in that central cavity that help stabilize the 2,3-BPG allosteric inhibitor, okay? So if we decrease the H plus, remember, so that means we're increasing the pH, so we're becoming more basic. These histidine side chains are going to become deprotonated because we keep going even higher of a pH above where they want to be when they're ionized. So then these become neutral, and then this molecule, the 2,3-BPG, doesn't really bind as well. So it like leaves way easier. And so that causes a stabilization of the R state. Okay, because remember, when that molecule is bound, it stabilizes the T state. Okay, fetal hemoglobin. So fetal hemoglobin has a higher affinity for O2 because it has, instead of alpha 2 beta 2, when you were a fetus in your mom's womb, you had alpha 2 gamma 2 subunits. So you don't express the beta subunits until you're after delivery, and the fetus no longer kind of gets the pregnancy hormones from the mother, and that switches up some of the genes that start being expressed. So especially in the form of hemoglobin, you no longer make the gamma subunit, you end up making the beta subunit, which is what all adults have. But the gamma subunit is different than the beta subunit from one amino acid, and that is a histidine to a threonine point mutation. And that's one of these histidines right here. So it's either this one, I don't know, we could probably look at it in Pymol. It's either this histidine or this histidine right here. And that single point mutation doesn't allow for the 2,3-BPG to bind as well. So what that means is that the fetal hemoglobin has a higher affinity for oxygen, right? It's stabilized in the R state more. And that's important because the fetus isn't breathing oxygen like the adult body is, okay? The fetus has to get all of its oxygen from its mother. And so you have at the placenta, which is just a fascinating organ, where the fetal blood kind of meets up with the It doesn't cross, but on one side of the placenta, you got the mother's blood. On the other side, you got the fetal blood. And the oxygen molecules transfer from the mother to the fetus because the fetus has a higher affinity for the oxygen. So it kind of steals it. Doesn't kind of, it definitely steals the oxygen molecules from the mother's hemoglobin. And so then that takes the oxygen molecules to the growing and developing fetus. Okay. So fetal hemoglobin, since it has that single point mutation, it's more of the stabilized R state. So it's that left shift of the sigmoidal curve. So it just binds oxygen better. Okay, so those are all the reasons why you can get a left shift. Now let's talk about a couple of reasons what would create a right shift of this binding curve. So this means reduced affinity. You're stabilizing the T state, so the deoxy state. So kick off oxygens, more oxygens from the 4 hemoglobin binding sites. Okay, increase the temperature, kick off oxygens. You increase the concentration of 2,3-BPG. You're gonna increase the stabilization of the T state. If you increase the H+ concentration, you're also going to stabilize the T state. So remember, increase H+ means that you're lowering the pH. So we've got more H+ around. Okay, so let's start first with it there. So how does an increase in the concentration of H+ cause T state stabilization? And again, you just point, bing, right here to these histidines because they have a pKa that's around 6 and blood is about 7. So if you increase the H+ concentration, you're going to make sure more of these histidines are protonated. And if you're making sure more of these histidines are protonated, that creates an even more positive cavity for this super negative 2,3-BPG to bind. Okay, so you can almost say that it's held tighter by those ion pairs. Okay. One thing maybe to think about is that at physiological pH, all 4 of these histidines are not necessarily all protonated all the time. Okay, the pKa is around 6, and in the future chapter we'll see that you could actually figure out what the exact pKa is about those. But if they're about 6 and you're at pH of about 7, that means that 90% of those histidines are already deprotonated. So only about 10% of these histidines would have a positive charge on it, right? So if you increase the H+, you're going to take that from maybe 10% being charged to 20, 30, 40, 50, 100%. So you are just creating that more positively charged cavity to allow for 2,3-BPG to bind. Okay, so high altitude. Why do athletes train at high altitude? Training at high altitude actually affects the concentration of 2,3-BPG in your system. So when you train at high altitudes or you go up to higher altitudes, it takes a couple of days, but your body will start producing more 2,3-BPG. So you're increasing the concentration of 2,3-BPG. But why? Why is that? Why does your body start doing that? Because it's acclimating to the new partial pressure of oxygen that's in the air. Okay, so most of us live around sea level. So on the y-axis of this graph, I have altitude, I guess, in meters. And on the x-axis, I have the total barometric pressure in millimeters of mercury, which is the same thing as torr. So if you recall at sea level, that should be 760 torr at sea level. As you increase in altitude, the barometric pressure decreases, right? At really high altitudes, there's lower pressure. So what does that look like in the air? So maybe at sea level, so air always contains about 20% oxygen, okay? But the pressure is different. So that the pressure has to do with kind of the density of the air. So at regular sea level, given the pressure being around 760 torr, for every, you have like 7 molecules of oxygen to 28 molecules of nitrogen. As you increase in the altitude, you're decreasing in the pressure, which means there's just less molecule. The density of the air is less. So and when you get up in higher altitudes, like at the top of Mount Everest, well, a little below that, but top of Mount Everest is about 8,800 meters. There's only like 2 oxygen molecules available. So down here at sea level, you were breathing in about on average 7, but when you move up in altitude, you're breathing in less. So your body has to account for that change in the partial pressure of oxygen at those higher altitudes. Let's look at a different graph. So here's another graph. On the y-axis over here, I've got partial pressure of oxygen in millimeters of mercury, or torr. And on the right side of the graph over here, I just have percent saturation of O2. We're not going to look at that. That's the red line. Don't look at that. Let's look at only the black line because it's kind of the same axis we've been working with, except for we flipped it, the X and Y. My x-axis is the altitude. So at sea level, the partial pressure of oxygen is about 100, and that correlates with the graph in your book. Like the lungs get about 100 millimeters of mercury of oxygen. That's partial pressure. Recall that your tissues are about 40%, around 40%. So as you increase in altitude, your lungs, instead of being saturated with about 100 torr of oxygen, I keep going up. Where's Denver? Denver is roughly the mile-high city. It's about 1 mile up. Denver, if you're breathing in the air at Denver, instead of maybe down here in Kentucky where we're breathing in 100 torr, you're breathing only 80 torr of oxygen, okay? That's your starting point for your lungs. Your tissues are always still gonna be about the same. So you can't deliver as well the oxygen. What's that look like on the curve? Here's a great curve that I found. So here we are with that percent saturation, or the fraction bound, on this y-axis. And here we have on our x-axis that partial pressure of oxygen. So remember, at sea level, it's about 100. So let's focus on the black curve for right now. So the black curve is just your normal, you were living at sea level for a long time, hemoglobin binding curve, right? So in the lungs, you're at about 95% saturation. When you move down to the tissues, which is about this 40% value, you're at roughly like 60% saturation. So you're offloading about 35-ish% oxygen. So you're delivering that much oxygen to your tissues, okay? When you immediately go up to high altitude, day one, go to Denver, okay? Denver has about 80 partial pressure of oxygen. So that already has shifted your starting point down a little bit. So now instead of starting at 95% saturation, you're starting at like 85% saturation. Your tissues are still going to be here at this 40% saturation. So notice though, instead of delivering this difference of oxygen, you're delivering this difference of oxygen, so less. So instead of offloading, I think I was saying like 35%, maybe you're only offloading 25%. So it's not as efficient. So your body's going to adjust to figure that out, and it adjusts by increasing the concentration of 2,3-BPG. Okay, that creates this right shift. Okay, so after you're at high altitude for a couple days, this would be the new shape of your hemoglobin binding curve. So 0.3 on the graph is after you've kind of acclimated to the new partial pressure in the air. And then 0.4 is the amount of offload. Well, I guess, sorry, the change from 3 to 4 is the offloading amount of oxygen that your hemoglobins are delivering from taking the oxygen from your lungs down to your tissues. Okay, so it right shifts because it wants to maintain that this difference right here. I keep calling it the offloading, but it is because the oxygen's being offloaded into your respiring tissues, right? And then travels back up to your lungs and recharges again to be, I mean, if you're at sea level, about 100% saturated, but if you're at high altitude, this looks like around the 70, 75 mark here. Okay, and then it goes and it keeps offloading. Okay, so that's why athletes train at high altitudes, because in short, it increases their 2,3-BPG. Oh, let me say this. So when you train at high altitude and you move back to sea level, guess what? This is still going to be your curve for a couple days. So you train at high altitude, the next day you fly to San Francisco, sea level, to run your marathon, do your Ironman, ride your century on the bike, whatever it is that you were training for. Your hemoglobin still have this curve because your body hasn't figured out yet, essentially, oh, we can like knock down the concentration of 2,3-BPG. So the normal schmuck that was training at sea level the lung, you know, we're starting at this point here. The lungs is here, and they've got the black line as the schmuck training at sea level. So here's their lungs, here's their tissue, and their tissues are working pretty hard because you're going to be running, you're going to be needing a lot of oxygen. So that person can only offload this percent. I think, what did we say? It was like 35% of oxygen. But if you're the guy training at high altitude and you came here to run your race, you're the red curve. Your body hasn't adjusted back yet to look more like the black curve. So you're the red curve. So in the lungs, you're picking up here at the red curve and you're still offloading here. So look at how much more oxygen you're able to deliver to your tissues so they can create more energy so your muscles can keep on working and pumping those pedals or running that race. So that's why athletes train at high altitude, because they want that right shift, and then they go and use their newly formed hemoglobin right shift to go run their race at the lower altitudes. So they get even more oxygen offloading from their hemoglobins. Okay, so that's the end. So what specifically is recap? So remember, this is structure function analysis of hemoglobin. So what specifically about the structure of hemoglobin aids in its function? Woo. Okay, so remember, kind of secondary structure between myoglobin and hemoglobin looked really similar. You can superimpose those things and all the alpha helices line up pretty well. But one of the biggest differences is the tertiary structure is a little bit different because it just has different primary. It's got a different primary sequence. So you've got different interactions on the surface here, which allowed for the oligomerization of the 4 subunits to come together. So the quaternary structure is very different. Okay. So the quaternary structure allows for it to have cooperative binding. Okay. That's one thing about the structure that aids in its function. You can also just, if you want to talk about structure function, you could talk about how these hemes are bound. So it still has that hydrophobic cavity for the prosthetic heme group to come and bind, right? That's still a thing. What else? That's 2 off the top of my head. Oh, structurally, it has this cavity, right? So it's got that central cavity, and it has some positively charged lysines, histidines, and N-termini pointed at it. So you can put in allosteric effectors, either inhibitor, maybe an activator. We only talked about the inhibitor. But so that's another structure function analysis. Okay, there's 3 big ones right there. Maybe you think of a couple more, but those are the ones I can think of off the top of my head. All right, so hopefully you enjoyed and you know so much more about hemoglobin in your body and its function to deliver oxygen to your respiring muscle or tissue cells.